Hi, hello, welcome to the Physionic podcast. If you're watching the video version of this, you might notice that I'm outside, mainly because, I've been quarantined or self-isolating for the last 3 weeks or so, and frankly, I'm getting really restless. I don't know if anybody else is feeling the same way that I am, but I've had to force myself to go for walks and do anything and everything I possibly can, from going insane just looking at research and doing my PhD work and all the stuff that's going to end up sticking me and leaving me inside for the entire day without human contact. As a self-professing, I'd say largely an introvert, although I have extroverted tendencies, even for an introvert, it starts to get a little much after 3 weeks. But with that said, let me introduce myself real quick. I'm a PhD student in molecular medicine. I have my master's in exercise physiology, although that's probably not going to be that much of a factor when we're talking about this particular topic today. And let's see. I've been doing cell biology research for quite some time. Cell biology, molecular biology research for quite some time. So the topics that we're going to be discussing today are, I'm going to be answering a few questions that I've received, some pretty understandable questions. So the last podcast that I released was talking about conspiracy theories. I'm not going to be going into those conspiracy theories anymore, although I certainly encourage, if you haven't listened to that one, it's like an hour-long podcast, so it's pretty long. You can listen to how I would explain some of the theories that people have come up with as rebuttals for if the coronavirus COVID-19 is man-made or not. My belief is that it is not, based off the evidence that I've shown in previous videos. But that said, I did want to cover a few questions that were understandable questions. Some people were just curious how I would answer these particular questions. So I'm going to jump into 3 of them, and then I'm going to touch on if face masks are an effective technique for reducing your risk of infecting someone else. So this is not going to be looking at if you will get infected if you wear a face mask. And this actually comes out from a study that I looked at. And originally I was going to make some unique content to this, specifically on that study, but the study is trash. And I don't know if the CDC is basing their recommendations based off of this study. I know they recently released recommendations, maybe a couple days ago, on people wearing face masks where they can't socially isolate. And I have to say that if it's based on this study, I don't feel that confident in that recommendation, which is a bit of a shock. But I imagine that there's other research out there as well that they've likely looked at. But this particular paper has come out in the last 3 or 4 days. And like I said, I was originally going to make specific content towards this, but it just didn't pan out that way because, frankly, I was just so blown away by how bad the paper was. But I'll go into that paper. So maybe I won't necessarily answer if masks are beneficial or not, but I'll at least touch on the subject based on the data that I'll be showing. And I'll actually throw up the graphics of what I'm talking about when I'm talking about the paper, because usually for the podcast I don't do much editing, but this time around I might as well go ahead and do that. And the 3 questions I'm going to be answering this time around before I get into the paper are: does SARS-CoV-2, which is COVID-19, have a patent? That seems to be a reoccurring question. Some conspiracy theorists have brought it up, but then other people have just been curious about, what's the deal with this? So I'll touch on that. Why is there similarity between SARS-CoV-2, which is again COVID-19, and the original SARS-CoV, which is of 2003, in symptoms and structure? And that's, I guess, the line of thought there is that it's proof that it's man-made because it has so much similarity to SARS-CoV, the original SARS. And why is this virus happening now? So we've had these wet markets for such a long time I don't know that much about Chinese culture, but I imagine that they've been happening for more than just 1 year. They've been happening for likely hundreds of years. So why is this coming up now? It seems too good to be true in terms of, China's and America's difficulties in terms of the economic struggles and things of that nature, and kind of the animosity between the two countries. I'm not going to touch on the politics of it because I'm not educated on the politics of it. But what I can explain is those 3 questions. So I'm going to go ahead and jump into the first one. So does SARS-CoV-2 have a patent? The answer is no, it doesn't. The reason why people keep linking this patent and they say, okay, well, this patent was made in, I don't know the exact year, it was like 2016 or 2018 or something like that, which is clearly just a few years ago. So meaning before the COVID-19 pandemic occurred. And they cite that as potentially proof that, this might be man-made. And it's not proof, because the patent is for SARS-CoV. So it is not for SARS-CoV-2. Again, I really want to make that distinction because I want people to understand that there is a big distinction between SARS-CoV and SARS-CoV-2, which is COVID-19. So this patent is for SARS-CoV, the first one in 2003. And the reason why they're using a patent is because they're looking at, I looked at the patent and it's just skimmed over it, looking at a gene sequence. So the patent is for a vaccine. So when you're developing a vaccine, not that I have developed vaccines myself, but just from my knowledge so far in my education, when you're developing a vaccine, you're sometimes taking different parts of a virus. So you're not going to necessarily take the entire virus and inject it into people, even if it's a somehow disabled version of that virus. You're going to take a piece of that so that you have different antibodies that can be created by your body to attach to that piece. So if they recognize that piece of the virus, then they'll attach to it and then essentially block it from entering your cells. Now, this vaccine is for a particular section of the virus. So they have decided, and the reason why it's a patent in this situation, I don't know if it ended up becoming an actual patent, if it was granted patent rights or anything like that. That's, again, outside of my area of expertise. But what I can say is that you can have like a particular section, and they outline that section through the genetic sequence. So that's why you see that genetic sequence. And that's why they have this patent, because they're trying to create a unique vaccine that's based off of that genetic sequence, which would be different from other vaccines that have already come out. I don't know. I haven't looked into this to that degree. But if there are other vaccines, a question people might come up with is, well, if we already have some, then why would we even bother having another vaccine? It seems really fishy. It's not fishy at all, because we typically have several different vaccines. If you want to make something more potent, have a better vaccine, a vaccine that's going to be 98% effective as opposed to 85% effective. Maybe it won't have as many side effects. Whatever it might be, like. It could be any number of reasons, just like any treatment that you might give, if that's a drug or whatever it might be. So in this situation, and I don't know the company, but whoever decided to patent it, labeled it under a vaccine. So most likely they're going to be taking that genetic sequence and creating a vaccine based off of the protein structure that genetic sequence encodes. So that gene will be read, and then it will produce a protein, and that protein will typically go as a target is a part of that virus. So then if you just express that particular protein, that's not going to do anything. It's not an entire virus. It's just a piece of the virus. So then from there, they can then create the vaccine and presumably inject it and see if antibodies will actually be produced to that particular sequence of DNA. So that's the reason. Is it SARS-CoV-2? It is not. So I don't want you to think that they've been developing this. This is somehow proof that they've been developing this in a man-made laboratory. So, or man-made in a laboratory. So why is there similarity between? So actually, this brings kind of to our next question. Why is there similarity between SARS-CoV-2 and SARS-CoV in symptoms and structure? So these are both coronaviruses. There are actually many coronaviruses, and this is something I'll touch on again in the next question. There are multiple coronaviruses, but the big three that we're talking about right now is SARS-CoV. People have mentioned MERS as well, and SARS-MERS, and then the other one is SARS-CoV-2. So the thing is, like scientists have named these viruses because they are similar, and people are taking that information and reversing it on the scientists and saying, you guys created it. If that logic doesn't exactly pan out. The reason why it's called SARS-CoV-2 is because it has similarity to SARS-CoV. That does not mean it's 100% similar, but it is very similar. And the reason why it has to be very similar to be called SARS-CoV-2 is because you're looking at a family of viruses known as coronaviruses. The term corona comes from the spike protein, and I mentioned this in a video. I didn't mention this particular aspect, but the spike protein is the part that will bind to our cells and then allow them to be internalized. So the spike protein will bind to our ACE2 receptor on our lung cells and then be internalized, or the virus will be internalized, and then the contents will be released specifically. So that spike protein has a crown-like structure, and that's why it's called a coronavirus, because some scientist was like, wow, that looks like a crown. Okay, let's call it coronavirus, and we're just going to fly with it. So the reason why those two are similar is because to get that crown-like appearance in that spike protein, you have to have a lot of similarity, at least in the way that the protein is folded, and typically in a lot of the gene sequences. And then it goes beyond that as well. So I imagine that there are a lot of different sections of that virus are very similar to the original coronavirus, because that's just the way it turned out. Otherwise, it would not be a coronavirus. So that's why there's a lot of similarity between SARS-CoV and SARS-CoV-2. And in terms of symptoms, it's just another thing. Like, I think the exact same argument could be said of something like influenza, right? If you have influenza A or influenza B, and then you have all these different mutations of influenza, which is flu, that happen over the years, we're just going to keep calling it influenza because it is largely the same as the other influenzas, or one other influenza, but it has mutated in a small way to make it different. So it's going to be largely the same, let's say 95% the same, but it will have a slight difference, and that's what makes it a different virus. That's all it is. So we wouldn't be calling it flu if it didn't give us fever and body aches and things of that nature, and it didn't affect the cells that it affects. Then it would be called something else, and then it wouldn't have that similarity. So there has to be similarity between a previous iteration of a virus and another iteration. Now, of course, if you keep mutating over and over and over, thousands and thousands of times, eventually you'll have certain differences, but still there's going to be some base or foundational level of this virus that's going to be similar to the future versions of the virus. So it's just not proof. It's not proof of anything. It should be that way. It just makes sense for it to be similar to the old version of the virus. So hopefully that gives you some explanation. I'm sure if you weren't convinced before, you may not be convinced now. But if you are on the fence, hopefully that offers some explanation. Okay, the third question is, why is this virus happening now? which is somewhat understandable. You know, under the current climate, I can understand how people might think that this was released because of what's happening in the politics and in the world right now in 2020, which, again, I'm not going to speak on. But why is this virus happening now? So the short answer is that it isn't, which is a weird answer. I can imagine you're probably like, well, what is this guy bloviating over? In reality, it's happening now just by happenstance. It just happened now. Not because of any particular design for it to happen in this particular time period. And the reason why I can say that is because there are many, many, many coronaviruses. It's not just this one. We're focused on it because it's a human coronavirus. It's affecting our population. But if you were to ask the bats, or if you were to ask the rabbits, or you were to ask the kittens out there, they would be like, well, where were you in 2016 or 2012 or 1998 or whatever it might be, because they have their own coronaviruses that don't affect us, and therefore we don't talk about it, but it does affect them. And it can be really deadly as well, for whatever reasons might be out there. So we're focused on this one right now because it's affecting us. We're being selfish about it, which is completely understandable because we don't really care about any other viruses. I mean, maybe if you have a dog and you really care about your dog and it gets sick with a coronavirus, of course you're going to care. But for the most part, we care about our own species. So that's why it's happening now. And it's always happening. It may not be mutating to a point where it's getting people sick, but this crossover between viruses and this constant contact between viruses happens all the time. It's happening millions and millions and millions of times, and it just takes one time. It takes the perfect storm one time for something to happen. And that's true of anything. That's true of anything in the entire world. If I can't think of anything off the top of my head, but like you could talk about like weather, for example. If a house is built to resist water and it's built to resist fire, but it can't resist both, and everybody's like, oh, it's really unlikely that it's going to happen at the same time, and then it happens at the same time, the argument suddenly isn't, oh, well, why did this happen now? It just happened. That's it. That's all there is to it. So I realize it's kind of a piss poor example, but my point being that this is happening now. It's not for any particular reason other than it was a perfect circumstances for it to happen. Constant contact leading to an eventual evolution of this virus, and other viruses were evolved at that same time. Don't get me wrong. Like, I don't want you to think this one virus popped up and that was it. That was the only one that popped up. It wasn't. There's hundreds, thousands of other viruses that popped up, but none of them could affect us because they didn't have the right pieces to affect us. But those viruses, if they had come in contact with a lizard, may have killed that lizard with 100% lethality. You just don't know. But for us, it was completely benign because it couldn't get inside of our cells. So it's happening all the time. It's happening right now as you're listening to this. It will happen forever into the future as long as viruses exist. Which, by the way, somebody said that all viruses are man-made. No, that's not true at all. A perfect example of that is smallpox, which is thousands of years old. And unless you think that science, which has not been around for thousands of years, science has been around for about 500 years since the scientific revolution and the Renaissance era and things of that nature. So unless somehow people use witchcraft or something to create viruses, then all viruses are definitely not man-made. That's just asinine. Okay, so hopefully that answered your questions. Those three questions that were kind of the big ones that I saw that had some sort of reasonable rationale behind them, like curiosity that did make sense that I wanted to address. And now I'm going to move on to this paper. Now, I'm not going to go into every single detail of this paper, but I did want to talk about this one aspect of this paper that I found, well, one, worrisome, and secondly, I just thought that the way the data panned out was just complete crap. And again, if you're watching this, if you're listening to this, I will have it linked for you, of course, as always. But if you're watching this, I'll throw up one graphic so that you can look at, what I'm talking about as well. So the paper is actually in a pretty reputable journal in Nature Medicine. So I was really surprised. I think they probably rushed this to a degree because, although I guess I can't say that, because the data, the study itself has actually been ongoing for 2 years, for a little bit over 2 years actually. So it's not like they just made up the stuff. It's been going on, and then this happened with COVID-19, and they probably thought, well, this is the perfect time to go ahead and try and knock that out as much as possible so that we can actually release this and have it actually be beneficial to the public. So the paper title is Respiratory Virus Shedding is Exhaled Breath and Efficacy of Face Masks. So again, I will have that linked for you. And let me quick go to the data. So what they did is they identified people over a number of years, 2-3 years, something like that. And they identified people that were suffering, that were in a hospital, so they already had the illness, the infection of the common cold, which is a rhinovirus, typically a rhinovirus, or the influenza, so flu, or coronavirus. Now, just to be clear, this coronavirus is not the coronavirus we're talking about here. This is actually a series of different coronaviruses. One of them that they did measure was SARS-CoV, so the first coronavirus, along with some other kind of smaller, lower tier coronaviruses that haven't really made a name for themselves because they just haven't been that terrible, although they do exist. So they lumped all those together, all the coronaviruses into one category, and then all the influenzas, and then all the common colds. And then what they did was they ended up measuring for 30 minutes of just regular breathing, how much virus is shed. So when our cells end up taking up a virus and start producing a lot more virus, they are shedding. They end up releasing a lot of virus, and then we end up breathing, or we can breathe that out, as we'll maybe see. So we end up breathing that out, or we cough, or we sneeze, or whatever it might be, and that is in that fluid. So when we sneeze or cough or even breathe, what that process is called aerosolizing. So where you aerosol, it's like air. The virus is on minuscule droplets. And so this paper ended up stratifying those droplets between over 5 micron water droplets, so a larger water droplet, or a finer water droplet, which most likely would be able to float longer, so under 5 microns. And the virus is going to be in both of those. And they wanted to know, okay, is a mask going to actually block that? So first they measured without the mask, and then they measured with the mask, and they wanted to know what difference there was. Now, they also did nasal swabs, and they did mouth swabs, or throat swabs, I should say. Now, I'm going to, again, put this up on the screen, but I'm going to first look at the coronaviruses, and that's probably what I'm going to be focused on. So with the coronaviruses, they showed that with the nasal swab, so each one of those dots, so there's a series of dots, if you're just listening to this, there's a series of dots that are on a graph, and they're red. And each one of those dots indicates a single point. Of course, dot point. Oh, wow, I'm really using a lot of synonyms here. A single measurement of an individual with this disease, with this illness. So in this case, the coronavirus, you should see dots across the entire graph, meaning that they're picking up some level of virus. And all they're doing is just measuring how much virus is there. And so the very bottom of that graph is 0, meaning they're not detecting any virus. And then as you go up, you're obviously getting more and more virus actually being detected. So with the nasal swab, you see all the participants had virus. The throat swab, which is right next to it, you see that most of the individuals have virus being detected. However, you do see that 4 individuals do not. They're flatlined. They're right at the bottom, so there's undetectable amounts of virus. So that's all fine and dandy. Okay, so here you're getting reading, with decent amount of reliability that these individuals have virus. So it's in the nose, we know that, and it's in the throat. We got that. Okay. Now, the next one over, they're looking at particles, again, the aerosolized stuff, the droplet particles of over 5 microns, or a micrometer, without a mask. So they're just breathing for 30 minutes, and they just want to know how much virus is actually being detected. And here you see three people that they're seeing, that they're measuring virus. And what is that, like 5, 6, 7 people that they're not detecting anything? So they got 3 people to detect a virus that's coming out of just breathing. And with these individuals, they were allowed to cough. So if they had to, not that they were incentivized to cough, they were told, please just breathe normally and we'll measure from there. But if you cough, don't stifle it. It's perfectly fine. And so with all of that taken into consideration, only 3 people showed small amounts of viral infection, or small amounts of actual virus. Then, so that already tells you that's pretty bad news, because I digress. I'll move on to the next section, then I'll explain my point. So the next one right over is the comparison, the exact same experiment where they're measuring with breathing over 30 minutes, but with a mask, a regular mask that you would normally see covering their mouth. And with that mask, they're seeing no detectable virus. So they're not detecting any virus escaping beyond the mouth. So it's just being stuck to the mask. Or is it? That's the question, because like I said, there were only 3 people that actually showed virus that was being expelled from the mouth. So how can you actually say that the mask is working if you only have 3 people that are expressing the virus, and the majority of the people that are being tested aren't showing any virus escaping their mouth without a mask? So then if you add a mask, that's not going to create much strength in terms of your experiment. So I found that absolutely astounding to me that they would even this is bad. This is just bad. So it's flatlined. Every person, they didn't measure any sort of viral release with the mask. But that doesn't really tell you much about the actual mask being effective, because it's possible that the mask may only be effective if a person coughs, but may not be effective when a person's just breathing, because when they're breathing, they're not actually releasing any virus. Or another reason is because they didn't measure long enough, or maybe, and that's probably a lower tier reason. I don't really care that much about that. 30 minutes is a pretty long time. However, going even beyond that, though, still is it's possible that the researchers just sampled incredibly badly, meaning that they screwed up the sampling over and over and over and couldn't get a reading because they didn't know how to do it, or they weren't doing a good enough job at it. So, but assuming that's not the case, which that's a big if, but assuming that's not the case, and they are detecting virus, they are doing their due diligence as much as they possibly can, 3 people is not enough to show that, or to convince anyone that without a mask, you're actually getting virus that's being exhaled out on a normal level. And then with the mask, that it's somehow knocking that down. So it seems to be really, really selective if that's the case, which I just didn't buy it I just don't buy it. So the p-value for that was 0.07, meaning that it's trending towards significance, which I think is a bit laughable if you actually look at the data, because again, it's just 3 individuals versus 0. So not exactly very strong. Then they looked at under 5 microns, so water droplets that could potentially pass through the mask, right, depending on the type of mask. And I think that's probably the big takeaway here, is that the type of mask matters very much, because if you have a mask that has big holes in it, I mean, you're not going to actually have a mask with big holes in it to the detectable eye, but if it's woven in a way that that allows gaps between each fiber of that mask, then that means that you're going to have bigger and bigger particles that can get through that. So if you cough, it's just going to spray right through the mask. The mask isn't going to be beneficial. So that is something that we can potentially at least caution ourselves with based off of this paper. But they looked at water droplets that were less than 5 microns, and here again, they're finding 4 individuals without a mask. 4 individuals show a certain amount of certain amount of virus being exhaled, and they're leaving about 6 or 7 individuals with no detectable signs of virus. And then when they use the mask, they find everyone has no detectable signs of virus. So again, is it because of sampling error from the researcher, or is it really because these individuals are breathing out virus and the mask is actually capturing that virus and keeping it contained? I mean, they did show that as significant, a p-value of 0.02, which again, I find that rather laughable, because it's not convincing in any regard. So that was coronavirus. If you're interested in influenza and rhinovirus, I will briefly touch on those. Again, they found the same for influenza and the rhinovirus, again, the common cold, with the nasal and throat swab. So those were consistent across all 3 different viruses. And then they, for the influenza, for the flu, they found a significant difference between wearing a mask and not wearing a mask. But the not wearing a mask had a total of 6 individuals expressing virus or exhaling virus, and they had maybe like 12 or 13, 14 people that flatline, just didn't have any viral expression. So again, not all that convincing. And then unfortunately, they had one person that did show an expression or a release of virus with the mask on. So again, and everybody else was flatline, showing no viral load. So not exactly very convincing, but they did apparently show that it was statistically significant. And then oddly enough, maybe not oddly enough, this makes some sense, but with the flu, they had more people. They had 1, 2, 3, 4, 5, 6, 7. I said 7 or 8 people that did release virus without the mask at less than 5 micron, lower micron or smaller water droplets. But then with the mask, they also had 6 individuals that did also end up releasing virus. So that implies that the mask is not very effective when it comes to the flu specifically. And finally, for rhinovirus, again, I said nasal swab and throat swab were the same, or showed higher increases. Although again, there are still a few people that didn't have any sort of virus, so that calls that into question a little bit. But for the most part, they did see increases. And then they found no statistical difference for the large microns, so the large water droplets. They're finding that for the droplet particles that are over 5 microns, they're finding that a mask does not help. They're seeing 6 people have virus escaping through the mask, even though they're wearing a mask. So, and it was even worse for when the water droplets were even smaller. So, I mean, look, folks, once you look at that data, it's not convincing at all that a mask will be effective. I would say on a rational level, it makes sense for a mask to work, of course, depending on the quality of the mask. And I'm assuming that the CDC is using other data that I would still need to investigate. I think my dad sent me an article wherein they talked about how prevalent just breathing, the amount of COVID-19 specifically, how much that gets expelled from the lungs just by breathing and talking. And so that actually hasn't been published yet, though. It hasn't been peer reviewed, therefore it hasn't been published, but some people had early access to it so they could look at some of the preliminary, like what the researchers ended up concluding. However, if this paper is any indication, you definitely need to look at the data yourself, which I've always been a huge proponent of. You need to look at the data yourself and not just base it off of the conclusions of the researchers. So once that paper comes out, I will hopefully get my hands on it. I will be able to get my hands on it, and hopefully I'll have enough time to actually go through it and read it. And yeah, that's what I've got for you. Masks, I would say, obviously still follow CDC guidelines, but if the CDC is using this study, I'm not convinced. However, wearing a mask isn't exactly going to hurt you, so it's not exactly a terrible sacrifice to wear a mask when you're out in busy public. Okay, so that's what I've got for you. Hopefully you found this informative. Again, really coronavirus, COVID-19 focus, but that's just the way things are going. I do hopefully have a really, really cool podcast episode coming out next week that is not going to be coronavirus focused. So I will tease that now, and if it happens, which I think it will, then you'll get to look forward to that, hopefully. Okay, well, thanks for tuning in, and I will catch you in the next one. Bye.